physiological impact of nanoplastic on the pulmonary circulatory rhythms and cardiovascular operations. As you know, currently we are facing the contamination of the nanoplastic or microplastic. So, in the environment, due to the industrial waste or other waste, we have reduced density of the nanoplastic in the environment. So, when you inhale the nanoplastic, how those nanoplastics actually interact and causes the damage of the lung. So, in this study, we have analyzed those activities and results. So, this is the overview of the presentation. So, as you see, the nanoplastic or microplastic can be found from the personal care product, then from the industrial waste, the plastic products, and textile products. So, these are the name of the common nanoplastics, the polyethylene glycol, PTP, PVC. So those are the name of the common microplastics that is found in the environment. And this is the powder form of the nanoplastic that can be inhaled and goes to the deep lung. So and this is the different structure. This is actually the polymer of the subchemical component. So the concept we have taken. So these are the nanoplastics. When you inhale, it goes to the deeper. When it goes to the deeper lung, we have the alveoli. So once it is entered into the alveoli, how this alveoli around interface can be impacted by those microplastics. So here I want to show some how those aorta interface, how the lungs affected will actually form. Once we have the lymphocyte type 2 cell in the lung, it produces some layer which is known as the lungs affected monolayer. So this is the lungs affected monolayer. When you inhale the particle, it actually interacts with this lungs affected monolayer. So this is the schematic structure of the lungs affected monolayer. At first we have the monolayer. Under the monolayer we have the bilayer. So how those bilayer and monolayer will be interact by the microplastic, we have to investigate here. And in our lung we have some lipids, DPPC, POPC, POPC and surfactant proteins. There are 4 types of surfactant proteins. About 8% of the surfactant proteins in there. Those proteins and lipids actually controls our inhalation and exhalation breathing. As we know, the surface tension of the aorta interface is 72 milliliter per meter. At the absence of lung surfactant, we have the high surfactant pressure. But in the presence of this surfactant, this pressure actually reduced to 22 or 25. At the end of the inhalation and exhalation, it goes to the 0. So, how those lipids and proteins control the normal breathing? Once we have the plastic or microplastic in the cell, it actually causes some damage. And this surface tension and pressure cannot be equilibrium stage. And the patient actually experience the shortness of breathing. Even in some cases, they can be died. So, this is the mechanism. Once we inhale the nanoplastic, actually, this is the deep lung. At deep lung, we have the alveoli. So, this is the alveoli air sac. And then, this is the first barrier and this is the monolayer. And we have the cell layer here. Once we have this nanoplastic, it passes through the cell layer. Before entering the cell layer, how this nanoplastic has caused the damage of the lung surfactant. So, this mechanism I will studies in my study. So, we have two air sac in our body. In the absence of lung surfactant, we have huge external pressure that can be collapse of the alveoli. Once we have the surfactant, there is an opposite force that actually equilibrium from 70 to 20 to 25. So this is the schematic. Once you introduce the surfactant, then the surface tension reduces to 20 to 25 at the end of the inhalation. And at the end of the exhalation, we have nearly 0, not exactly 0, nearly 0. And this surface tension and pressure can be equilibrium, and the people can take a healthy breathing. Once this is changing due to the nanoplastics, then this surface tension can be varied and the patient will be experienced the shortness of breathing. So these are some literature review that have motivated us to work on this field. So here they have just conducted the experimental studies. So the authors will use experimental first, but I'm going to do the competition before doing the experiment. So these are another study on this field. So after literature review, I have set up my research aims and objectives. So nanoplastic penetration mechanism, how the nanoplastic interact enter the cell, then that determine the toxicological impact. How amount of the nanoplastic may cause the damage of the lung circulation. Then the clinical inhaled nanoplastic concentration, the effect of the air compressibility for healthy breathing, and analyze the viability of the nanoplastic absorption of the aerobic process. So there are two types of study. We have the experimental arm and the simulation. I'm working on this part. My experimental class has been conducted by Poznan University of Technology, Poland, and another part has been conducted our pharmacy department in Joshua Scientific University. So this is the protocol, how I have prepared my model. So this is the model. This model, in the absence of nanoplastics, once we have add the nanoplastics, the model look like this. So now I'm talking about what is molecular dynamics. So all the computational calculation has been done by the molecular dynamics simulation. What is that? This is a computer simulation for biological molecules. So here we apply the very popular equation, the Newton's second law of motion, but in the nanoscale, not for the micro or meter scale. So we have F equal to mf. F is the force, m is the mass, and it is the acceleration. So we have acceleration as the second order differential equation. We can solve by using some mathematical equation. And we have the force. Force can be balanced to the potential. So the gradient of the potential, we can balance those equations. We have the final potential energy equation like this. So we have the potential energy due to the moment, the potential energy due to the angle between some molecules atom, and the dihedral angles for 4 atoms, and we have the non-boundary interaction potential, that is the linear adjusted interaction potential, and we have the Coulomb potential for the charged particle. And we have done this simulation by using Chromax. So the molecular dynamics simulation can be done in lamps for material, and the Chromax can be done for the biological molecules. And the visual molecular dynamics software I have used to visualize the system. So this is the protocol and how to solve this equation. This is also the mathematical equation. The first one can be solved by the Euler equation. This is very well-known equation to solve the ordinary differential equation. And we have the elliptic algorithm by using the non-linear method. In order to solve this equation, we need around 7 terabyte memory for completing a single study. So this is the typical calculation that I have done in my pictures. So this is the area for lipid. What actually happened there? So we have the lipid. The lipid has two heads. Once we increase the surface tension, we monitor the area for lipid. That is when the area for lipid is lower, that is at low surface tension, the lipid molecule actually get the perpendicular. And also we have the higher area for lipid, the lipid actually reduce the water parameter. So this is the water parameter. Also increase the plastic concentration, the water parameter is decreasing. Why? Because the surface area is increasing. That's why the lipid gets more space to sit in there. And also analyzes the mean square displacement to show the dynamics of the nanoplastic, how the nanoplastics will causes the movement of the lipid molecules. So I calculated the MSD, that is mean square displacement for the lipid GPC, POPC, POPG and cholesterol, and I found this result. I have also calculated the MSD, and I have also calculated the diffusion coefficient by using the Einstein diffusion equation as well. So this is the and here, as you say, once at the initial of the simulation, the nanoplastics actually interact with the layer. At time, those nanoplastics penetrate in the membrane. How this membrane actually changes with time. At the end of the 600 nanosecond, I found that it produced the bilayer for the downlayer and the mycelium.
For the time limitation, we cannot continue anymore. Please.
Thanks. So this is the outcomes. Once we complete this study, I will solve the answer of this question, and this is the potential influences.